All right. And then get started. Okay. So when we left off, we were talking about our different classes of biomolecules. So we've gone through our carbohydrates with their carbon, hydrogen, and oxygen, polar hydroxyl groups, oxygen and hydrogen together. And we had talked about our lipids, which are characterized by being non-polar and hydrophobic generally. And we mentioned our specific, most important type of lipid, the phospholipid, has amphipathic, so it has that polar head and that hydrophobic, non-hydrocarbon part at the tail. All right, so we have those two properties. We've got the lipid hydrophobic part and this special group making one end of it hydrophilic. And we talked through our proteins. So we mentioned that proteins are built up of amino acids, contain a carboxyl group and an amino group, so contain nitrogen, particularly characteristic of our protein. We talked about their levels of structure. So we talked about primary, secondary, tertiary, and quaternary structure, so building peptide bonds, and hydrogen bonds, subunit interactions, and then R group interactions, and finally subunit interactions to build a whole big functional protein that can do lots of stuff in your body. Okay, so that means we have one final group of biomolecules next. So our last group of biomolecules that we're interested in are the nucleic acids. So the monomer of a nucleic acid is called a nucleotide. And so it goes along with the name. And we can see parts of this structure that are familiar. So if we're looking at an individual nucleic acid, so looking at one nucleotide, we're going to see, hey, we can recognize that we actually have a carbohydrate or a sugar ring here in the center. Right? So we're not going to get fooled by that because there's more stuff. But we can already see that we're based on a carbohydrate here. Now looking at a phospholipid, but we can see we also have a phosphate group here, right? So that might be familiar from our other type of biomolecule as well. But we can see what's different is that we have a nitrogenous base here. So B is not for an element, that's just telling you this is a base. And we've got a bunch of different choices for our nitrogenous bases that may be attached at that location. Okay. So our nucleic acids have this basic structure. The two most important nucleic acids we'll be talking about are DNA and RNA. They're hopefully familiar in previous courses, but a quick reminder of how they differ. So when we're looking to tell whether we've got DNA or RNA, which type of nucleic acid we're looking at, one of the things that we can key in on if we have the whole structure is what type of carbohydrate we have. So what is forming that ring structure at the center, okay? So RNA is made of ribose. So ribonucleic acid is what that stands for, right? So ribose, plus it's a nucleic acid, it tells us in the name. DNA is made of deoxyribose, so deoxyribonucleic acid is what DNA stands for. So we can see the difference in those two carbohydrates down below. Okay. I want to point out, we're going to actually start with the ribose. All right, so here we have the ribose structure. So we've got this carbon ring structure. We have a lot of hydrogens and hydroxyl groups coming off the side, right? And we're going to focus in on the bottom, right? So our ribose has an OH here and an OH here. So that's our general structure for ribose. What's different to recognize deoxyribose is we deoxygenated it, right? Deoxy, we removed an oxygen. So if you look over at the structure of the deoxyribose, we've still got one little OH here hanging out the bottom. But at that second position that we were examining, we have only a hydrogen, right? So we removed one of the oxygens on the structure of this carbohydrate ring. So that's how it gets its name. Otherwise, they look the same. So ribose for RNA, deoxyribose for DNA. Now, often when we think about genetics, when we're thinking about nucleic acid, we may not draw out the full structure of the carbohydrate that's at the center. Often we'll get like lists of base pairs. So those bases, so those nitrogenous bases, where we have our options, we've got cytosine, going to be abbreviated C. Right? We got thymine, abbreviated T. We've got uracil, U. And we've got adenine, A, and guanine, G. So these are grouped together based on their structure. So you can see a reason these three are at the top. These are the pyrimidines. They've got just one ring in their nitrogenous base. Our purines have these two rings pressed next to each other. Okay. So when we're looking at our RNA and our DNA, we might be paying attention to which of those nitrogenous bases we have. Because if we have uracil specifically, that's going to tell us we've got RNA, right? At least that's the one I like to focus on. Pretty obvious. Otherwise, we have pairs that we expect either in RNA or in DNA. So we're going to have cytosine and guanine, thymine and adenine in our DNA. Now, we're not going to talk a ton about genetics in this course. So just remembering nucleic acids exist is for the most part what you're going to need as you move forward. But I do want to point out some molecules related to these nucleic acids that we've grouped together with them that we are going to talk about a fair bit in this class. And those are our energy transferring nucleotides. So this you may have focused on the left hand side. I don't know what you did in your chemistry classes. Okay, but I'm going to point them out. Okay, so in particular, we've definitely talked about in terms of its name is ATP, right? If you haven't seen ATP, you should come talk to me. We need to get you up to speed on that. And we're going to be talking about that directly kind of next. Okay, what ATP is adenosine, so adenine and ribose, plus 3 phosphate groups. That's where it gets its name. So we can see the adenine structure, right? The ribose sugar in it, right? So that's our adenosine. And triphosphate groups will tell you there are 3 phosphate groups attached, right? That's what the T in ATP is for. So you can see those here. So 1 phosphate group, 2 phosphate group, 3 phosphate group. Okay. So what you're looking at down the bottom here is an ATP molecule. This is going to be fundamental for understanding how cellular processes work, because we're basically always making ATP so that we can shuttle energy to different places in our cells, break apart the ATP to use energy to make things run. The way we use energy is we break bonds, right? So bonds between different atoms contain energy in them, right? So when we break them, we release the energy in the bond, and we can use it to power other processes. So we have a picture of adenosine triphosphate here at the bottom, but we're going to have an important reaction where we break and form a bond. So we're going to be breaking apart our phosphate groups, right? So it says 2 or 3 phosphate groups. because adenosine triphosphate has three. So this is our form of the molecule that has lots of energy in it that we can use. So when we use it, we pull off this final phosphate group, right? We go pull that apart. And that leaves us with only two, right? So only two phosphate groups here are left on the molecule. And so that's our adenosine diphosphate. So di is for two, right? So that's our ADP. So when we talk in our understanding of metabolism and cellular respiration next, about ADP and ATP, right? When we're doing metabolism and we're trying to make energy in the form of ATP that we can use, we're going to be putting in ADP, adenosine diphosphate, so this low energy form. And through the process of breaking down molecules, breaking down glucose, we're going to form an additional bond. We're going to trap that energy in that additional bond, forming ATP that we can then take somewhere else in the cell. So that's why it's worth particularly noticing this structure. This isn't the only time we'll see it. This isn't the only time I'll say this. We'll also come back in muscle. This is our preview here. You could have adenosine monophosphate too, with just one attached, but that's not going to be particularly important for our energy transferring processes that we'll be talking about in this course. Another example of an energy transferring nucleotide that are a little different is NAD. So we're going to see nicotine adenine dinucleotide as well when we talk about metabolism from here. But that's also based on this adenine structure, right? So similarly, you'd be able to find this somewhere in its structure, just like you could find that in ATP. We've got different groups attached to it. This is going to be shuttling electrons around in our metabolic processes so that we can make ATP kind of heavy. So we'll take a look more specifically about that when we get to those portions of metabolism. Bladine adenine dinucleotide is also going to be working the way that NAD does. So these are all versions of nucleic acid kind of built on this nucleic acid nucleotide structure. Other nucleic acid derived structures that we'll see will be our cyclic nucleotide. We won't get super deep on this, but you will see them, particularly cyclic AMP. Cyclic AMP or CAMP, right? Cyclic tells you it's a cycle, right? So you can see that our phosphate group here is like a circle, kind of. That's how I remember it anyway. So we've got this phosphate group forming a ring. And these are going to be used as cellular messengers. So when we talk about pathways in cells or how receptors work, we're often going to be talking about a cell senses something out on the outside of the membrane and then produces a signal inside the cell, usually based on cyclic AMP, to send responses throughout the internal parts of the cell. So that's where we'll see that type of nucleotide come back. Okay. So those are our four types of biomolecules that we're interested in. So we got our carbohydrates, our lipids, our proteins, and our nucleic acids. And now we're going to start to be moving toward our understanding of metabolism. So we're going to dial in and focus in on our enzymes, our type of protein. If you're trying to link this up to your textbook, this is actually at the beginning of chapter 3, rather than in the section on biomolecules specifically. Okay. So since we're understanding enzymes in order to understand metabolic reactions, let's review for ourselves first this concept of activation energy. Okay, all right, a little bit split, which is fine because we're about to draw a graph. But when we're thinking about activation energies, we're going to see that an activation energy is something we kind of have to overcome for a reaction to happen. So if we have high activation energy, that means we have to overcome kind of a lot. So our reaction is going to happen more slowly than if we only need a little bit of activation energy, in which case it's easy to overcome. And that's going to make our reactions go faster. That's the gist of it. The reason I was asking you about that up first, because the point of an enzyme, point of the specialized type of protein, is to lower activation energy and to speed up reactions. So we're going to look at activation energy in pictorial form in a second, but what we want to know first is that our enzymes are proteins. They speed up chemical reactions, they increase their rates. This means that we refer to them also as catalysts. So that's more of a chemistry word. Right? Catalyst is something that does this. So catalyst is making this reaction happen more easily. So from an energetic perspective, what's happening here when an enzyme acts as a catalyst is it's decreasing the activation energy for a specific chemical reaction. We're going to have lots of enzymes, and they're going to be different levels of specific. Sometimes we'll have enzymes that can do a couple of different things, but we've got so many different kinds because we have so many different chemical reactions that your body needs to do. But all of these enzymes are going to be working to control rates of reaction, right? And when you have the appropriate enzyme, it's going to speed up that reaction specifically. Okay. So if we were to write this out in kind of a chemical formula-ish way, right, what we would do is say that we're going to have to add in an enzyme and what we call a substrate. So a substrate is going to be the thing that an enzyme binds to. So in this case, with substrate, we're linking this up to your understanding of chemical reaction. It's going to be like a reactants. So substrate and reactants are mostly the same thing. And for a second, we're going to have our enzyme and substrate bound together. So here's that enzyme substrate complex. And then that enzyme substrate complex is going to break apart. So we're going to get our product, right? So our substrate turned into a product. And we get our enzyme back, right? So we get to reuse enzymes. We recycle them. They're not actually part of the product that we're making, right? So they don't really count as a reactant itself. They're the catalyst that makes that reaction happen. So if we're thinking about the activation energy barrier, right? So what we're thinking about is our reaction happening over time, or our reaction coordinate here on the x-axis. Reaction. And then we're going to be thinking about the energy it takes on the y-axis. This is our energy. So what we're going to be doing fundamentally in our reaction is we have our reactants that have some certain amount of energy, reactants, and they're going to turn into products, right? So in this case, I'm going to draw energetically favorable reaction, so one that might happen spontaneously. So here we see our product with less energy. But regardless of what reaction we're talking about, reactants are going to have a certain amount of energy, products are going to have a certain amount of energy. So the activation energy barrier refers to the fact that we don't just draw a straight line from reactant to product. Right? Usually both the reactant and the product, some amount of stability, right? Like they don't want to immediately change into each other. So we're going to need some kind of instability here in this system. So we might speed up the reaction, we might just jiggle it around, or it might just happen by random chance. But regardless, we're going to need to have a little extra energy in the system before we then fall away and get our energy in the product, right? So how high this lump is our activation energy. So the difference between that peak and the amount of energy we had in the reactants just on their own, that's our activation energy barrier. So at a minimum, this takes some amount of time, even if this is a spontaneous reaction, one that happens naturally. So what our enzymes are doing, right, when we say they're lowering the activation energy barrier, what we're saying is that in a reaction that has an enzyme present, so we're still going to have our reactants with that same amount of energy, but it's going to be a little easier to go to a product, right? So we're going to see that it takes a little bit less activation energy when we have an enzyme present, right? So that means we can speed up this reaction by adding an enzyme acting as a catalyst. So that's what we see happening here. So here's a picture in general with all this stuff labeled out, a little prettier than mine. You get the idea. So big activation energy without an enzyme. When we add in an enzyme, this gets a little faster. And this is good from a cellular perspective because we want to be able to control rates of reaction. So you can control the rate of reaction, adding more enzyme or by taking more enzyme away. We're adding and subtracting enzymes, controlling how much enzyme we have present. In order to speed up reactions, we've got a lot of enzymes. And we want some step of a reaction to be a little slower. We're only going to have a little bit of that. So this way we can have everything moving nicely in order along and not have to wait forever for things to randomly hit the activation energy barrier. We have a way that enzymes work, like from a physical perspective, that allows them to lower that activation energy barrier. You've probably seen a picture like this somewhere in your life before. What this is looking at is a physical depiction of how enzymes work. Okay. So you've got two models that we can use to explain how enzymes work. The typical one you learn first is a lock and key model. This works pretty well for understanding enzymes. It happens to not be perfectly accurate. There are some enzyme reactions that are reversible, that if you're really focused on the details of the lock and key model wouldn't make sense. So we also have an induced hit model with a little adjustment that works similarly to the lock and key model with a little bit of a twist. So in the lock and key model, what we're looking at is an enzyme. So here's the body of the enzyme. It's a folded up protein. We've just drawn it as a little blob because we don't care about the exact chemical structure for the purposes of this physiology course. But we do care that we've got this little pocket here. That's our active site. So our active site is where the substrate is going to bind in. So we have substrates, the reactants in our chemical reaction that act as substrates when they bind to an enzyme. Now, in this lock and key model, what we're saying is that that active site, that pocket, is not this little circle shape on accident, right? That circle shape because it fits like a lock with a substrate that's key shaped, so one that fits nicely into that pocket. So they're the same shape, they're attracted to each other. We'll see in a minute the word affinity, so I'll just mention it now. When we say something has affinity for something else, what we're saying is it's attracted to the thing, right? So these have great affinity for each other because they're the same shape. Maybe they have opposite charges attracting each other like little magnets, right? They mesh together nicely. That means they have high affinity for each other. And if these other shapes don't fit in as well, if they fit a little but not very well, we might say they have low affinity, right? So high affinity is good, means a good fit. Now, this other point about the reversibility of enzyme reactions gets to the fact that when our chemical reaction happens, right, we wouldn't be trying to take a little circular substrate C here and just turn it into another identical circle that is also substrate C. Right? That's not a product, that's just the same thing, right? So because some enzymes can take reactants and bind them and help them turn into products, but can also take those same products and do the backwards, turn them back into the original thing that we think of as a reactant. We need to tweak this model a little. So that's where our induced fit comes in. So in this induced fit model, what we've added here is that when a substrate binds into an active site, and so enzyme, active site, substrate, all that stuff is the same here. What we're saying is that we have a little bit of a shape change in the active site. And this is just true in chemistry in general, right? When things bind together, they change their shape, right? But this is certainly true for proteins, right? When they interact with each other, we know that their structure is all based on hydrogen bonds and R group interactions and subunit interactions, right? As you start to get other chemicals near those, they change a little bit, right? Like I'm working on all these like kind of pseudo-magnetic forces, right? So I'm gonna have a change. So here we have what we refer to as a conformation change. Conformation just really means shape here. Right, so we got a shape change in that active site when our substrate binds it. So we can see this enzyme alone in our induced fit model. That's kind of a square shape in that locked part. We can see when the substrate comes in, we have the shape change. And so the presence of that substrate actually makes this fit work so well, right? So now we've got a good fit because the substrate came near the enzyme and they bound together. Enzymes work partially by holding the substrate, holding reactants in a specific position. So if you imagine like a covalent bond as like a rubber band or something, like literally imagine it physically, you can imagine an enzyme as it changed shape. Maybe it pulls, right? Like maybe it pulls opposite sides a little bit apart, right? That's actually how this works if you're thinking about breaking something up, right? It puts the molecule into a very specific orientation. It makes it more likely that that reaction will occur. Okay. So all this to say, substrate bound to the enzyme. Because of the way the enzyme works as a catalyst, we get that substrate, that reactant turning into a product. You can see that this product is a little differently shaped than the substrate, right? So we went from circle to some kind of polygon here, right? Doesn't fit so well in this pocket that our substrate made for us. It gets released, right? So released out to the world. And now we have our enzyme back, open with an activation site, ready to do this reaction again, but we're ready to go. So the fact that we can have this slight shape change in the active site is why we could use an enzyme like this for a reversible reaction. Okay, so imagine that we have one shape change when the substrate binds in, but let's imagine that a product or maybe a different similar molecule came in and bound here. Maybe we'd have a different shape change. So induced fit molecule model just says that we have a bit of a shape change happening. Otherwise, it works the same as the lock and key model. So enzymes aren't the only thing that we'll see when we think about how chemical reactions work in your body, work in a cell. We'll also see these things called cofactors and coenzymes. So basically, they're going to be working along with our enzymes. So you can think of them as like little added groups. So our cofactors are going to be something that's not a protein, that have to work in conjunction with our enzyme for that enzyme to work and to have the right shape to work as a catalyst for a reaction, right? So enzyme specifically is a protein, right? So it's that specific type of biomolecule. So if your enzyme has to work with a bit of magnesium, or your enzyme has to work with a bit of copper, right? Some inorganic mineral or something, right? That's going to be a cofactor, right? Our coenzymes are different. So our coenzymes are going to be little chemical groups. So they may look like organic molecules, like little functional groups, organic functional groups attached to our enzyme. So they need to be present for appropriate enzyme function for the enzyme to work the way we want it to, but they're not their protein in their own right, and they're not directly like on the active site, right? We need them for the enzyme to have the proper shape and have the proper function, but they're not going to be directly involved in like forming and breaking the bonds of our substrate. So when we think about vitamins, right, we often need vitamins because they give us coenzymes. So that would be an example of a coenzyme. They often transfer small little chemical groups in chemical reactions as well. So these participate in the reaction in a slightly different way than an enzyme does. We're going to think a little bit about rates of reactions and amounts of enzyme as we start to think about metabolism and why we have these enzymes. So I'm going to answer this slightly intuitive question first. So if we add an extra enzyme to a reaction, add more enzyme, what's going to happen to that reaction? It's going to happen faster, slower, or is this not going to make a difference? Yeah, more enzyme working as a catalyst. That means that we can do this more times. We usually aren't thinking about changing just one individual molecule, one individual reactant into one individual product. We've got lots of versions of that same thing happening all at once. So when we have more enzymes, overall, if we're thinking about that reaction, it's going to happen faster. So when we think about enzyme reaction rates, we're going to be thinking about the enzyme's catalytic rate. So that's going to be how fast the enzyme itself works, basically. So how many molecules per unit of time that enzyme can help us transform. But we're also going to need to think about how much of the substrate is present, or how much of that reactant is present that we're trying to turn into a product. We're also going to need to think about how much of the enzyme do we have present? So enzyme concentration is going to make a difference. Then we also have to think about the affinity of our enzyme for the substrate. So how much the enzyme and the substrate like together, how fast they bind together, how fast this enzyme substrate complex forms. Because we're going to find that some enzymes are really, really attractive to the reactant that they're helping catalyze in this reaction, but some enzymes aren't. And we can see changes to this enzyme substrate affinity based on other factors like pH and temperature. So some of those regulated variables for homeostasis, some of the reasons they're regulated is because they can change enzyme function, change enzyme affinity for substrates. So we have temperature and pH down there at the bottom because those are particular ones that would mess with this whole enzyme system. So we're going to dial in specifically on that affinity piece. But we'll mention some of the other things as we go through here. So we're gonna just draw in general our reaction rate now. So this isn't energy anymore. We're just talking about how fast does this overall reaction happen, right? So now times kind of over here on the y-axis. And on the x-axis, we're looking at substrate concentration. Okay? So we're thinking about how much substrate is present. So how many of those reactants are present. So one thing that's true is if you have more substrate, so more reactants, so long as you have enough enzyme, that's going to raise your reaction rate, right? So substrate concentration goes up, as long as there's enough enzyme to catalyze the reaction, our reaction rate goes up. Now, eventually, we're going to level off at some point, right? So at some point, we're going to hit a maximum reaction rate. This is going to be dependent on the function of that enzyme, and we're varying the amount of enzyme, how much of that enzyme is present. So we do hit a maximum reaction rate. We can't go faster. We found all the substrate, all the enzyme that's present. There's kind of nothing left to do. So that's our baseline for substrate concentration and reaction rates. Now we're going to add this affinity piece on. So if we think about this reaction, but we're going to think about an enzyme that has low substrate affinity. So an enzyme is like, it'll bind to that substrate, but it doesn't really like to so much, right? Like it'll do it, but kind of slowly, kind of weakly. We're going to see a difference in how that reaction rate and substrate concentration are related to each other. Yeah. So we're still going to see that our substrate concentration as it goes up increases the reaction rate. It's probably not going to do it as quickly, right? So it can bind to the enzyme, but it takes longer, doesn't do it as well. So it's not going to speed up the reaction rate as much as a substrate that had high affinity for that enzyme. May also be the case that the maximum rate, depending on that affinity, different enzyme, different substrate affinity, it might max out at a different kind of level too. Okay. So this depends on exactly why it has that low enzyme substrate affinity, but kind of low, high. So slower reactions when we have low enzyme substrate. So we can regulate our enzyme activity as well. So we may want to do this because when we're thinking about metabolism or other cellular processes, there are often lots of steps. Probably remember that from your discussion of metabolism in other courses. Right? It's not just one step. Right? And we don't want to have kind of overproduction, a really speedy part at the beginning, and then we've got a lot of product that's kind of like waiting around and maybe diffusing around the cell because we're waiting for there to be enough enzyme or enough functional enzyme to do the second step. So we want to regulate these steps so that we're making the right amount of product and we're kind of matching that to the amount of reactant that we expect to have. And there are a couple ways we can do this. So the first, I like to think of these in kind of two chunks. So the first two are going to be kind of physically, how do we do this? How do we change how the enzyme works? And then the second two parts are going to be more kind of conceptual. So like when we're thinking about negative feedback and positive feedback, these aren't the same thing. They're not the same thing, but conceptual that way. We're thinking about pathways. When we're thinking about math, we're thinking about maths when we think about these second two parts. Okay, so we're going to start with our kind of physical changes to enzyme activity. So the first one called allosteric regulation. So allosteric, this is, I think, coming from Greek roots. Allo means other, steric means place, okay? So in allosteric regulation, what we're saying is that we have some other molecule, we're going to call our modulator here. So some modulator, some other molecule that's not the enzyme, not substrate, binds to some other place, allosteric, on the enzyme, and that changes how the enzyme works, that changes its function. So you can see how that works on this depiction here, right? So we still have an active site on this image, right? And we kind of left it the same shape. We left the enzyme the same shape as our example on the previous slides. So we still got an active site. This is where our substrate would go, right? So substrate needs to go here. But you can see that's not the only part of this enzyme that does anything. Right, so at the active site, where our substrate is binding, where the reaction is being catalyzed, but we also have this regulatory site on the other side. So the square regulatory site on the top is where we see this modulator binding in. Right, so this is our modulator bound into the enzyme. Okay. And because when different molecules touch each other, they change shape, right? We have this conformation change. The effect of this modulator binding into the regulatory site is coming in contact with the regulatory site is that we change the shape of the active site. So the part that actually does something, the part that actually binds into the substrate, the part where the reaction actually happens. So you can see that this active site is now a different shape down below, which is going to change how well that enzyme binds to any given substrate, right? So this is going to change the affinity of the enzyme for a substrate. So it's going to make it either soak up that substrate more quickly, make it easier to bind to the substrate, or make it harder. And it could go either way, depending on what that modulator is, depending on what this enzyme is. And so this can make it more functional or less, speed it up or slow it down, but it's definitely going to change it. So that's our allosteric regulation. Our second piece is our covalent regulation. And this is similar, a little different. The difference is going to be we actually form a covalent bond. So in the allosteric regulation, you see that this is just kind of sitting in the regulatory site, or it's kind of sitting in this pocket. So if we have something just sitting in that regulatory site, but not actually forming a covalent bond where we're like sharing electrons, right? We're just got a nice fit. We've got some R groups that are attracted to whatever that modulator is. So we got this free kind of interaction. That's great. But then that modulator will also release from that regulatory site relatively easily. But if we have covalent regulation, we've got a more complex system and we actually form a covalent bond between our chemical group that's changing the activity of the enzyme. This usually has to happen with another enzyme helping out. Okay, so this is often what we're going to see when we think about like our coenzymes, right? So in this case, in our covalent regulation, we got our enzyme, it's got its active site, so still the circle and down on the bottom here. And we're still thinking about this regulation happening fundamentally by changing the shape of that active site, right? So this is why it counts as regulation. This is why we're changing the activity. It's still changed what's actually going to happen to the substrate of reactants or products. But how we change that site is different. Okay. So here, what we did, we tacked on an additional chemical group to this other part of the enzyme. So we wouldn't think of this quite as modulator and regulatory site. We're going to be specific that this is covalent regulation because we specifically formed kind of like permanent chemical bond here, sharing electrons with our additional chemical group. So this effect is going to last until we break that bond. Okay, we're not going to have that chemical group just like peel off at random. So we're going to usually have another enzyme, two enzymes, one that helps us form that covalent bond, and then another that breaks that covalent bond when we want to release this chemical group. So this is more useful when we want to change like long-term activity of an enzyme. This is more of a permanent change. We can delete it with an additional enzyme, but until we break that bond, it's going to stay there, stay attached to our enzyme, stay changing its activity. So particularly common ways of covalent regulation have to do with changing phosphate groups. And we have specific types of enzymes. So these would sub in for like enzyme A and enzyme B that do this. We're not going to focus in on this particularly in this course, but if you've ever heard of protein kinases, right, those would be adding one of these covalent bonds. So we're going to add together a new covalent bond between our enzyme and this phosphate group, right? So that's a covalent bond here, not a minus sign. And our phosphatases are going to break those bonds, giving us just a clean enzyme without that additional old chemical group. So that's now separate. So the plus sign telling you that's separate. Finally, that gets us to these kind of pathway ways of regulating enzyme activity. So we're going to have our feedback inhibition first. So what we're thinking about here, both with our feedback inhibition and our feed forward activation. So we're thinking about how we change the speed of steps, right? So we're not thinking exactly about like the shape of one specific enzyme, although that may be involved. We're thinking about how do we make sure that this first reaction, A to B, is happening at the same speed as B to C, as C to D, so that we get just one nice smooth flow down the line. Okay. So the way this works is that we can have a situation, feedback inhibition, where one of our products, so in this example, C, this product C, this intermediate product C, right, is itself like a modulator of the enzyme of either the step that produced C or one of the earlier steps in the process, right? So we're turning A into B, we did that with an enzyme, B is now our substrate, binds to enzyme 2, makes us product C, but then once we have C, if C acts through like allosteric regulation, let's say, on enzyme 2, going to change the rate of enzyme 2, right? So changing how this piece works. So in this case, when we're thinking feedback inhibition, it's changing it, and specific, this minus sign tells us it's slowing it down. Feedback inhibition is when we have a product or an intermediate that itself decreases the enzyme activity of that step or one of the earlier steps in the process. So this helps us when we are trying to match up our rates so that we can have a nice chemical pathway where everything flows smoothly. So this would make it so that we're producing a lot of our enzyme C, or sorry, a lot of our product C, intermediate C, we'd slow down that B to C step, right? So making it a rate limiting step by slowing it down, right? So that we can smoothly go straight to D. Here's another example. Doesn't have to act just on the step itself. Could go all the way back to the beginning. And we'll mention feed forward activation at the beginning of Friday, but basically it's super rare. So you're going to guess an answer. The answer should be feedback inhibition, but you do want to understand those.